My name is Isak Ingerholt, and today we heard many talks about the amazing applications that we can use wood for. But in my project, I'm trying to make sure that we have plenty of wood in the future. In Sweden, we are lucky that 70% of our land is covered by forest, and most of this is production forest. And it's essential that we make sure that we have a productive and sustainable forestry in the future, but this comes with many challenges. And one of the main challenges are fungal pathogens called heterobacillium. These pathogens are rotting trees from the inside out, and in Sweden, the primary host is Norway spruce, and it's estimated that 15% of all the spruces have stem rot upon harvest in Sweden. And when the fungus enters a tree, for example, through a wound in the bark, the tree will respond by trying to restrict the pathogen to only a small area. In the inner bark, the green here in the figure, the tree will start a necrosis. Here, the tree sacrifices a small part of the tissue while filling it with harmful chemicals to try and slow down the growth of the pathogen. Meanwhile, at the edge of the necrosis, a barrier will start forming. And this barrier we call the ligno-suberized zone. And the tree must form this barrier before the pathogen reaches it, otherwise the pathogen will break through and continue spreading throughout the tree. And in my research, I'm studying a few spruce genes that we hypothesize are involved in ligno-suberized zone formation and in resistance. One of these genes is called LAC5, or in short, LAC5. And this gene we know is associated with resistance to these pathogens. We know that it becomes very active when the tree is attacked by heterobacillium, and we know that it's involved in lignification. And lignification is something that takes place during formation of this ligno-suberized zone. And recently, we have discovered a rare variant of LAC5 that enhances resistance towards these pathogens. But now we want to pin down the role of LAC5 and understand what makes this resistance variant special. So for this, we use mainly two types of plant material. We have identified and collected spruce individuals that carry this rare gene variant, and we are genetically modifying spruce to increase LAC5 expression. And then we follow the ligno-suberized zone formation in these plants using microscopy. You can see an example here in the figure. Microscopy, gene expression analysis, and chemical profiling of the ligno-suberized zone. And an important goal is for this to have practical implications in the Norway spruce breeding programs. Because if we know the genetic traits that are important for resistance, we can precisely select for these traits in the breeding programs. And this will help our trees adapt to current and future challenges, and this will also contribute to a productive and sustainable forestry. Thank you.